This is a production of Cornell University.
All right, so I am the last speaker of the session, and I'll introduce myself. For those who don't know me, I am Olena Vatamaniuk. I am faculty at the Soils and Crop Sciences section and Plant Biology section here at Cornell. And what I'm going to talk today about is about using synchrotron X-ray fluorescent microscopy. I'll refer to it as XFR, as an imaging and high-precision phenotyping tool for studies of mineral nutrient homeostasis. And I'll talk today mainly about iron, micronutrient iron and micronutrient copper, well, because that's what we study in the lab, and also because of the relevance of these elements for the future food security and human health. Just briefly what I do today, after a really short introduction providing the rationale for studies of iron and copper, homeostasis in plants, I'll briefly introduce the basics of synchrotron X-ray fluorescent microscopy and the relevance of this technology for studies of mineral nutrient homeostasis. And then in the last part, I'll show you some examples from my lab which demonstrate how XRF was useful for us to understand the function of the oligopeptide transporter OPT3 in iron homeostasis and the whole pathway, SPL7 regulatory pathway, SPL7-SET1 regulatory pathway in copper homeostasis and relationship between copper and plant fertility. And again, the studies are done mainly in Arabidopsis, but I'll continue with some crop species. So, as many of you appreciate, providing enough food for the growing world population is one of the top challenges of the 21st century. And even today, up to 1 billion people in the world go to bed hungry. They consume less than 2,000 calories per day. And these are people mainly in the developing countries that you can see from this map. But, and in these countries, people mainly, because of their resource poor, they mainly confined to plant-based foods. And so these plant-based food, the staple crops which are grown and used in these countries, mainly rice, corn, and in some cases wheat, they inherently low in the concentration of nutrients and minerals such as iron and copper and zinc. And so in addition to consuming fewer than 2,000 calories per day, people in these countries, they suffer from so-called hidden hunger, which was the term coined by Ross Welch here at Cornell at USDA to reflect the nutrient deficiency associated health disorders. For example, many of you know, and many of you probably know that iron deficiency anemia is one of the most prevailing nutritional disorder, and it affects more than 30% of the world's population, and actually it is responsible for up to, well, 1 million deaths annually. And again, if you look in this map, these are people which are living in developing countries, and again, people in these countries are confined mainly to plant-based foods. And so one of the ways how to combat this malnutrition is to biofortify crops with nutrients and with minerals. And this emerges as one of the sustainable approaches how to deal with malnutrition. What's happening here? Okay, so copper deficiency in humans is not as widespread as iron deficiency. However, copper deficiency in soils can really dramatically decrease crop productivity, while it significantly affects plant fertility, and wheat anecdotally suffers apparently the most from copper deficiency. And those deficiency, like iron deficiency in the soil and copper deficiency in the soil, and then it's actually zinc deficiency, occurs mainly in alkaline soils. Here in the map, you can see it colored as blue. And these alkaline and also natural soils, they occupy more than 30% of the arable land. All right, and so if we really want to sort of meet the challenge of providing increasing food production by 70% in the next 40 years, and also if we want to make sure that we are able to biofortify edible parts of plants with nutrients, we really need to know how crops deal with their environment, the soil environment, and the limitation of micronutrients in the environment. And so it comes down to knowledge of the transport systems. So we need to know how which transporters and which regulators of those transporters are responsible for moving elements under deficiency condition from the soil into the root, how these animals, how these elements are distributed in the plant body, loaded into the edible tissues, how they are reabsorbed, for example, from leaves and moved to sinks such as seeds. And in that regard, X-ray fluorescent microscopy, it emerges like really powerful tool which allows us to identify novel genes and characterize genes which are involved in mineral nutrient homeostasis. For example, the reason why XRF is so useful and can be useful, because normally, in many cases, disruption of genes encoding transporters or regulators of the transporters will lead to changes in the distribution of the spatial distribution of elements in a plant, in a tissue, or actually and subcellularly. And so one can just use XRF to compare a wild-type tissue versus the mutant tissue and see whether the changes of the spatial distribution occur or not. And here, this is almost a classical example from Mary Lou Gorilneau lab showing Arabidopsis wild-type seed and iron distribution in wild-type seed. And this is a mutant for the vacuolar iron transporter. And I guess it's a mutant seed, and you can see that the pattern of iron distribution significantly changes. And of course, combining other functional genomic approaches, one can infer what is the gene function. So how XRF helped, what's XRF, first of all? So XRF is an imaging technique which allows you to visualize the spatial distribution of elements in a sample at the different resolution scales. And really, in a nutshell, very simplistically, XRF is based on the detection of secondary arrays, or we call it fluorescence, which are emitted by atoms of the sample when the sample itself is rastered through with X-rays, really high energy X-rays that are generated at a synchrotron. And the really cool feature about this technique is that each element has its very specific energy fingerprint. And what it allows you to map the distribution of, you basically can follow the distribution of particular element in a sample. And here it's exemplified by the distribution analysis of different elements, as you can see, calcium, copper, zinc, and iron in the leaf, and that has been done simultaneously. So basically, the advantages of using XRF over other imaging technique, well, first of all, it's incredibly sensitive, right? So we can visualize micronutrients such as copper, which is present in plants in really, I mean, it basically is single digits ppms, and in mutants even below ppm values. So we can still do that. Also, the excellent feature to us of this technique and to many is that it's not destructive. So you don't really need to prepare samples, well, depending on the resolution you want to study. So we can just take what we do in a lab. We just take a leaf, go to the synchrotron, and look at the elements, which is amazing. It's very high spatial resolution, and we can analyze pretty much all elements of the periodic table. And for us, important elements are copper, iron, and we're also leaning more towards zinc. So how this technique helped us? So, we were working on a transporter called oligopeptide transporter, which was initially identified in the Gary Stacy lab. And it has been known that OPT3, I'll refer to it as OPT3, oligopeptide transporter, is somehow involved in iron homeostasis, but the substrate of the transporter was unknown. And the OPT3 itself, the mutant, has very intriguing properties, of which I'll just want to highlight one. Leaves of the OPT3 mutant accumulated significantly, like huge amount of iron compared to the wild type, but yet seeds of the mutant contain significantly less iron than the wild type. It was unusual because typically seeds get their minerals from leaves, and you would be expecting that if leaves have so much iron, then seeds would have so much iron, but yet it didn't happen. And so to understand like what's going on with iron in the leaves, we decided to use XRF. We want to see well maybe iron is distributed differently in the leaf of the mutant versus the wild type. And we collaborated with Mary Lou Gorilneau, and what we found that actually iron is trapped in the vasculature of the leaf of the mutant. So what you're looking here is the iron distribution in the wild type, and it's mainly, you know, it's evenly distributed, but with main localization in vasculature. But then when you look at the leaf of OPT3 mutant, you can see that iron is highlighted in the minor veins, okay? So it's trapped there. So it already led us to the idea, well, iron just cannot be remobilized. So that's why it's so high in leaves and it's so low in seeds. And of course, we moved on to see, well, vasculature consists of xylem and the phloem. Xylem, that's the vascular tissue which delivers minerals from the roots and into the shoots, but then phloem is the tissue which is responsible for immobilizing minerals and delivering minerals from the sources such as the leaf to sinks such as seed. So we analyzed the iron concentration in xylem and the phloem sap, and we found that actually the xylem sap hyperaccumulates iron, while the phloem in the mutant has significantly less iron. So basically what we are looking here is iron in the xylem. Okay? And so next question was, okay, well that's the mutant for OPT3, so what OPT3 is doing? And so the next step was, well, we have to know where the OPT3 localizes. Is it in the xylem? Is it in the phloem? Because that can tell us what it does. Is it moving iron from the xylem, and then the mutant hyperaccumulates it in the xylem, or it somehow moves iron from the xylem into the phloem, and so it cannot load the iron into the phloem. And so just simplify the story, basically we found that OPT3 is localized to the phloem. So the phloem transporter, and mutation of which leads to the decreased concentration of iron in the phloem. And then the final sort of cherry of the cake was actually finding that OPT3 is actually an iron transporter, and we collaborated with Miguel Penaros from USDA here, and we showed together that OPT3, when it's, well, what you're looking here is oocytes, Xenopus oocytes. So we did some uptake studies in Xenopus oocytes, and so OPT3 is actually localizes to the plasma membrane, and it can, in Xenopus oocytes and in Arabidopsis protoplast, and it can move iron into the oocyte cells. So what you're seeing here over time, OPT3 is black bar, and you can see that it has OPT3 expressing oocyte cell contain more iron than control non-expressing cells. So basically, this helped us to understand. So basically, XFR was sort of the first step forward to understand what's happening to the OPT3 mutant, and why it hyperaccumulates so much iron in the leaf. It cannot move it into the seeds, and basically it's because the iron is trapped in the vasculature. And of course you have to combine XFR with other techniques, what we've done, to fully characterize the gene function. And so what we sort of hope for, we hope that OPT3 and its homologs from crop species can be used for biofortification, because OPT3 is responsible for loading of iron into the seeds. But that's next step. And now I sort of switch gears to copper. And again, just to reiterate the point that while copper is not as, copper deficiency is not as widespread for humans, for the plants it can be detrimental. And in fact, well, I teach mineral nutrition, and I teach in a class that, well, copper is needed for fertility. And we know that's in a field, right? So you can see in a wheat grown under really low copper is infertile, and, you know, and yeah. But then we never knew why copper is needed. And so, but however, copper is toxic, together as iron is toxic when it accumulates in excess. So plants have to really tightly regulate copper homeostasis to make sure that it has enough, you know, to prevent deficiency, but also avoid toxicity. And so this regulation in the plants is achieved by the transcription factor, which belongs to the family of SPL or SBP binding domain transcription factors, SPL7. SPL7, what it does, like really simple model of the SPL7 based regulation of copper homeostasis. Under copper deficiency, SPL7 upregulates expression of copper uptake and the transporters, and also genes, encoding transporters, which are responsible for the internal copper movement in a plant. And the second role is that SPL7 upregulates the so-called copper reutilization system, or we call it copper economy system, when plants try to feed on their own reserves and just metabolize, just get rid of very abundant but non-essential copper protein, and so deliver copper to much more essential copper requiring functions. And so it is not surprised that SPL7 mutant, of course, is sensitive to low copper in the environment. And the point here that SPL7 was the only transcription factor which was known to regulate copper homeostasis in vascular plants. And so we were very excited when a PhD student in the lab, J.P. Young, she identified a novel copper-responsive transcription factor, which we designated CYT1, copper deficiency-induced transcription factor 1. And of course, the bread and butter work, right? You have to see whether, you know, it's upregulated, it's copper regulated, but whether it has something to do with regulating copper homeostasis. We always have to go down to the mutant. And so J.P. isolated the mutant, and she grew the mutant in the presence of low copper, and what she found that under control condition, the mutant was looking pretty much as a wild type, and here you can see this is the mutant expressing the CYT1 genomic fragment, so it's a complementary line. But then under copper deficiency, the mutant was growing really poorly, and also it developed these chlorotic spots on mature leaves, which are indicative of copper deficiency, and then the genomic fragment can fully complement the mutant. We also found that this, you know, copper's increased sensitivity to copper deficiency is associated with the decreased ability of the mutant to take up iron and move iron in a plant body. So, for example, here you can see that in a root, the CYT1 mutant has significantly less copper than the wild type, and it's pretty much true for each tissue. And also we analyzed flowers, because remember I mentioned that copper was associated with, somehow it was always mentioned that copper is needed for fertility, and so we also analyzed copper accumulation in flowers of the mutant, and it had less. And also what is noticeable here, that actually there was less seeds in a sleek. So yeah. And then next we decided to see, well, is there a relationship between this newly identified transcription factor CYT1 and the previously known transcription factor SPL7? And so our rationale was that, well, if these two transcription factors, they work in the same pathway, then in a double mutant, we should see something like SPL7-like phenotype, which is the smaller growth under limited copper condition. But that's actually not what happened. Initially, well, it was difficult for us to find the mutant. It was so tiny. It took us a year. Well, really, literally, it took us a year. We were looking for it. and that, you know, we had to have a magnifying loop, and we found it. Well, but the problem was that the mutant eventually died, so that was the reason. So the phenotype was seedling lethality. That was, it took us so much time to understand that actually that's an issue. And which meant that the pathway is much more complex, so that what we previously thought. But then we were able to rescue this seedling lethality of the mutant with high copper. So again, what you're seeing here, different plant lines, wild type, CYT1, SPL7, single mutants, and the double mutant. So the double mutant now grows up to the reproductive stage. Well, it's smaller, but it actually has the same amount of copper as wild type, all right? So that was already for us interesting, right? Well, it's smaller, but has the same amount of copper. But then what we noticed, that although the mutant, the seedling lethality is rescued, and the mutant grows, the double mutant grows to the reproductive stage, the double mutant is fully infertile. Well, it's 90% infertile. So what you see here again, and this is at 50 micromolar copper, which is really high. It's high non-toxic, we call it. So basically, you can see other genotypes are doing quite well, but the double mutant, you know, it's infertile. These are just elongated carpels. Really, to cut really long story short, we were able to detect that that's due to pollen defects. So the double mutant produces like 90% less pollen, and then of the pollen produced, more than 20% of the pollen produced is unviable. So it's very, very strong pollen fertility defect. But again, this double mutant had the same amount of copper as other lines, right? And that was for us very puzzling, like what's happening there, right? And of course we were thinking, well, maybe there are a couple of hypotheses. One of the hypotheses, maybe these two transcription factors, they correlate copper delivery to the specific flower structures, right? That was our favorite hypothesis. Although we could not exclude the possibility that these are transcription factors. They can regulate so many other things in a plant, and maybe it has nothing to do with copper. But we favor the first hypothesis, and so how do we do that? And that's how we turned again to XRF. We thought we have to really look where copper localizes within the flower structures. And I want to emphasize that it was not known before. What we knew before, it was just copper is needed. But where copper is it petals? Is it sepals? Is it anthers? Is it, well, no one knew before it. And so at that time, we already took advantage from the fact that we have synchrotron facility here at Cornell. So that's Cornell high energy synchrotron source here, Wilson Lab, and that's Bradfield Hall, okay? Very convenient. It just depending how you go, different routes, but regardless, 5 minutes at most. So the postdoc in the lab, Ju Chanchie, Hoatin Zhen, they collaborated with Rong Huang and Arthur Wall at Chess, and together what we were able to find is that actually the majority of copper, if you look at the flower, and this is the wild type, and what you're seeing here is the distribution, the copper density. The majority of copper is really peaks at anthers. It also localizes at carpels, but you know the majority is still in anthers. And then when we look at the different mutant lines, we can see that, well, SIT1 is, well, it's somewhat affected. SPL7 is more affected, but then the double mutant has barely detectable copper, all right? Then we also looked for other elements. We looked for iron, and I mean, it's a long story why we looked for iron, but it was very important to us to show that it's not just, so it's actually just copper, it's not iron. And that's what we are seeing here, that iron is almost no, I mean, it's not statistically significant, basically. And so basically what it allows us to show that copper, well, first of all, copper is localized in anthers and also in carpels. And then we can pinpoint that this pollen infertility defect of the double mutant is actually associated with the inability of the mutant to move copper into the anthers. All right, and just to conclude this part, so basically it was known for a while that well, flower development and flower fertility is controlled by many factors, which include photoperiod and hormones. So using XRF, we were first of all able to identify which floral organs contain minerals, and also XRF allowed us to put transition metals, and in this case copper, as very significant contributing factor to plant fertility. So where are we going from here? Of course, we are very interested now to translate this knowledge we gain in model Arabidopsis to crops, right? And so we are collaborating with Mark Sorrells. We are using mapping population, wheat mapping population generated in Mark's lab with the hope to identify QTLs which will sort of be associated with copper delivery to anthers fertility and improved yield of wheat. And what you're seeing, we already started doing this work. So what you're seeing here, you're seeing florets of two parental line and actually their offsprings. And what you're seeing, that two parental line, they have different, they differ in accumulation of copper in anthers. And actually, I don't know, incidentally, these two parental lines also differ in yield. I mean, Mark can correct me if I'm wrong, but they, and so it seems that the W7984 is low yielding variety, and it yet accumulates less copper in anther. And also what we are seeing that there is a segregation for copper accumulation in anthers and actually in all reproductive organs, because this is carpels. In the mapping population. So we really hope that this will lead us somewhere. We are also collaborating with Susan McCouch, and we are trying to sort of adapt this technology also to rice. And this is, we call it semi-high throughput. So these are 14 lines in 6 hours. So we're talking about high throughput. Yeah. So these are seeds of rice, and what it allows us to see the distribution of elements in seed tissues. And the importance of this is such that I mentioned before that rice is really, all of the majority of cereal crops, they have very low concentration of minerals in grains. But rice is the worst, and especially white rice is the worst. And the reason is that the majority of minerals, especially such as iron, they accumulate in a hull in aileron layer. And so the breeding should be, and all of the goal would be to actually to identify varieties that will have iron not, you know, and maybe zinc and other elements, not the iron layer N in the hull, but actually moving iron into the endosperm. And so this is like the first step forward, just as 14 after, I think, 2,000 lines that have been generated in Susan's lab. And to conclude overall this talk, I hope I can sort of convince you that XFR imaging is a really powerful tool, one of the tools that can help us to infer the function of genes in mineral nutrient homeostasis. And I really hope, and we all hope who use this tool, that it will help us to breed for better crops, for safer and more nutrient-dense crops. And with that, I would like to acknowledge my lab members. So Jepei, she was working on SPL7 Set1 project. Ju Qianqi and Huaxin Zhen, they are two people in the lab who are really pushing forward this XFR studies. And I would like to thank our collaborators, Arthur War and Ron Juan from CHES, Miguel from Robert Holly Center here at Cornell, Mark Sorrells, Susan Makuch, Mary Lou Guerino, and Leon Koshin, and of course the funding. And we have one question, or we can take it outside. And thank you very much for the attention.
This has been a production of Cornell University, on the web at cornell.edu.